if the particle is in the presence of other particle, then of course at some point the trajectory of one particle can become close to another, and you lose information about the identity of the particle. Okay, so another issue, so other than the time you can measure, which is very important because you can discover different phenomena depending on the length. The other important thing is also how you localize the particle. So if you can localize in 3D, one very simple method is simply to make a Z-stack, and then you measure that particle, that section, and maybe if you're lucky, you can follow the same particle in 3D. But remember that every particle is determined by essentially the diffraction limit spot, which is an area function. So, for example, one possibility is to look at the shape of you measure for one particle, and that in basis on that shape, you can determine, well, that will depend, of course, on the radio, will depend the maximum Z you can measure. And this table show you that the maximum Z you can measure is approximately a value of between 2.5 and 3 micron. So you cannot really, by using this technique, measure wherever the particle will go. So evidently, we need other methods to do the particle tracking. This is the particle tracking, which is the fastest one. And essentially, you have a particle which is trapped in a given position by a laser beam. And then the particle, for example, will fluctuate about that position. And of course, that gives you eventually the position within the trap. And the dimension of the trap, of course, is determined by the force you can apply and so on. And then by a quadrant photodiode, so a photodiode which is sensitive to the X and Y position, you can determine the position of the particle. And this is very fast. In general, you can go to kilohertz or something like that in this kind of thing. But clearly, we lack of a technique that this is only if you can trap the particle, so it has to be a relatively large particle, but we lack of a technique that can give us a general method. So let us see what can be the technique. So really, many years ago, we thought that if we can do a very fast image of a region of a cell, and then by very fast I means, but still we need to have only one particle in that position, then we can track the particle, and then that is essentially if you can make very fast images in one position. And this is very important because give us an idea of how fast you have to go and how fast you have to measure. But now let us think something completely different, which starts from a different principle. So when we illuminate the particle, if the particle is in the center of the volume of illumination, then you will have that the particle will appear as a Gaussian. So that means that if that is the center of the volume of illumination, the red line represents the position of the particle. So if the position is at the center, well, a movement in X and Y or Z will not give any change because the derivative is 0. But if the particle is in the region of maximum derivative, so in that position, a small change in the position will give a relatively large change in the intensity that you measure. And this is the principle that we will use. So the question is how to keep a particle which is in the maximum derivative point. Well, this is not possible because, as the particle moves, you have to be able to track, and you notice position to derivative only if you are able to measure the entire point of function. Otherwise, you cannot do it. So let us see how we can keep a particle in the position of maximum derivative. Okay, so that was the technique that we did with one of my postdocs at that time, Katerina Krispetykova, and let us see how it works. So suppose the particle is at the center. If the particle is at the center, and then we can move the volume of illumination, which is that shadow that you can see here, we can move all around. Well, if the particle is exactly at the center, we get no changes as we move around. But if the particle is in position out of the center, a given amount, for example, in red, when we examine the particle by moving the Boltzmann function in that position, it will appear very bright in this position, but when the Boltzmann function is in the other side, it will appear very, very low. So it will appear very high at some angle, but very low at some other angle. If the particle is, for example, in the green, so it's even farther from the center, then as we go to the orbit, there will be a point of very high intensity and another point of very low intensity. And that will depend on the radius of the particle and the radius of the orbit that you use. And it was already said that the best radius of the orbit is about 1/2 the resolution you want to obtain. So clearly you can see that the position of the particle to respect to an orbit which circulate around the particle can be obtained by simply measuring the intensity. So if the intensity is in the red, the particle would be found at this angle. And if the intensity is in the green, we will find at the same angle, but with larger intensity. So if we measure the intensity and the position or the phase of the orbit, then we will be able to determine the particle. And of course, in the Z position, which is there, which is completely different than the other, we should be able to measure the particle too. So you have seen that from this idea to measure the derivative is not possible to be used unless we move the orbit. But if we move the orbit, we can keep the particle, we can determine the position of the particle by when the particle will appear as you move the orbit. Of course, if the particle is very far, we will have some problem. But let us see how it's up. So first of all, this is a real track. So we have 360 degrees on the orbit, and then we have a phase, which will be whatever, 190 degrees, something like that. So from the phase, we can determine the maximum position of the particle. And the AC, which means the ratio between the alternate part, which is that alternate part, with respect, sorry. Oops, I did something wrong. Oh, well. I don't know what I did wrong, but I jumped at the end. So sorry for that. Okay, so I have to go back to my original thing. Okay. So, the point is that if we can orbit around the particle, then we can tell where the particle is. And this is something to understand, that the speed in which we can correct the position depends on the orbit, but the position of the particle, so that position of the particle is determined point by point along the orbit. So general point along the orbit can be measured in microsecond, and an orbit can be measured in a millisecond. So this is the way the method works. So that doesn't seem to be a 3D particle tracking, seems to be 2D particle tracking, but let us see the parameters. First of all, we can measure an AC to respect to the average DC, and that is called the modulation. And then the phase is just the arctangent of the B factor, so how much is the AC to respect to the DC. So this is what we can have. And so if we can have something like that, that means that we can orbit the particle. As the particle will move a given amount, we can recenter the particle by a mechanism of feedback. So the feedback is always on in such a way that we want to minimize the AC part. So because the AC will be 0 if we minimize completely, And the angle in that case will be undefined because the particle is at the center. Okay, now let us see what happens with respect to the DC, to the modulation. The modulation is a little bit more strange. So if the particle is exactly at the center, the modulation is 0, because as we go around the particle, no matter what is the radius of the particle, the modulation will be 0, because it will be, all the points will be equivalent. As the particle start to move from this position and start, for example, in that position, then we start to have a modulation, for example, at a given angle. And later on, so if we continue to move the particle, you will see that when we cross the orbit, that will be the maximum of the modulation, and then the modulation will continue to increase. This is important that the function of the modulation has that behavior, because if we minimize the modulation is minimal at the center. And remember that the farther we go, it remains constant. So this is important, because no matter how far is the particle from the center, we can minimize the position in order, or position of the orbit, in order to bring the position of the particle in the center. Well, I say that this is a 3D particle tracking. Where is the 3D? Well, we're doing an orbit around the particle, and we can, I don't know if you see my hand, and the orbit around the particle. But nothing prevents us to run the scanner in order to do an orbit in the other direction. So we do in one direction or in the other direction, or we can do a combination of two. We can do, for example, one orbit in one direction and one orbit in the other direction, and get the simultaneously, so we in two orbits, but it is essentially simultaneous, the position along the z, so the change in the z, and the position along the xy. And that is how this method works. So we will always perform like an 8, so one orbit in one direction and one orbit in the other direction, in order to get this information. And this is, for example, how experimental value for the modulation is gone. So as if you're out, the modulation will be very large. If you're out in the other direction, the modulation will be out. When you are exactly in the center of the orbit, the modulation will go to 0. And this is an experimentally measured. If you have a fixed particle and you measure the modulation function. So essentially what happens is the following. Now, no matter where is the particle, you will have either a modulation of very large, and then you will have to bring to the 0 position. And that will occur in the X and Y direction, X, Y, and Z direction simultaneously. So in some way, we can track a particle in 3 dimensions with this method. So actually, we do the following thing. So suppose this is the point square function and this is the particle. So we do an orbit, for example, we call up, and an orbit which call down. If the particle is exactly at the center, the up orbit or the down orbit will give the same intensity. If the particle is in the up direction, well, the up orbit will give a larger intensity. If the particle is in the lower intensity will be obtained when it's down. So essentially, the modulation is obtained by the ratio intensity up, intensity down divided by the sum of intensity up and intensity down, which is the formula for the GP. So essentially, 2 orbits, one up and one down, will give us the average position of the particle in the z direction. And so that is what is happening. In order to do that, we have to displace the orbit up and down. Now that is essentially is a tricky thing, because generally in a confocal microscope, you displace the z position by applying a voltage to a device, and so it will move up and down. Now that cannot be done in this method, because if you move down or you move up, it takes several milliseconds for the system to relax and to get to the position of the equal position from the top of the lens. And so we need a different method. A different method in the orbital tracking is obtained using what is called an electrical lens. So an electrical lens contains a medium which gets compressed, and that compressed or enlarged, and that changes completely the focus of the lens. For example, this is if you have a lens and you have a piezo. So with the piezo, you measure a response which is not very fast, but with the electrical lens, you measure a display which is instead very fast. And so that is real, and this is the response of the electrical lens. Maybe you can see here. So that it responds very well up to about a kilohertz. So below 2 or 4 kilohertz doesn't respond very well, and actually has even a resonance time. But you know this function, so you can compensate for this function. Okay, so this is the experimental setup. So the experimental setup, we have the objective, which is moved by a nanopositioner in order to generate an orbit up and an orbit down. And the difference in distance between the orbit is generally 100 or 200 nanometers. Then we have a laser that illuminates. So we have a scanning mirror which performs the orbit in the XY direction, and then a dichroic mirror which is in every microscope in order to obtain detect the fluorescence intensity in that position, and then you have a computer. So this is the way the system is made. Still, there is one thing, which is how you start, how you determine which particle you want to look. Okay, so first of all, you need to set the initial position for the scanner. And this initial position, so you do an image, and then you say, I want to track that particle I see there in the image. And then you determine the position X, Y, and Z as a function of time, and then you calculate the intensity profile and the particle position. Once you have the particle position, you feed back the value and move the scanner and the Z positioner in such a way as to be exactly in the center of the particle. So this is the way it works. And so this is now the program. And the program, for example, you say, I want to look at that particle, and then you click that particle, and the scanner, which is in this case, in this position, will go to the center, then we'll measure the modulation. The modulation has to be constant around the particle, and so on. And then you move, for example, if the particle move in 3D, in 3D, you can follow the particle moving in 3D, and then you get the 3D orbit here, a position of the particle. So that is the way it works. And so let us see now what other parameters we can measure and how we can determine what is happening. So first of all, one very important thing is that while we are orbiting the particle, we can measure the lifetime. The other thing is that we can measure the shape of the particle. For example, if the particle is round, will give a different effect in terms of the harmonics that you can find in the YZ plane, and that will give you the shape of the particle. So if it's elongated, it will be different and so on. And finally, we can obtain also the spectrum of the particle if we do the things like it will be described by Belen in the next lecture. So we can measure lifetime, we can measure polarization, we can measure the spectrum, and we can measure the shape. All of them simultaneously while we are chatting. Okay, so suppose we have, for example, 3 particles at equal distance. So as we go around the position of the particle, well, we will measure every position of the orbit will be more or less the same. And then we can determine, for example, the number of particles. One is here, one is here, and one is here. And that will correspond to the position to respect to the orbit. Okay, but now what is interesting is the following. So I said we can measure the shape of the particle and so on, but okay, there are some limitations. For example, photo bleaching, if the particle photo bleach, well, we cannot measure anymore. And this is an example of the photo bleaching of many particles. And you can see that you cannot measure anymore that because there is no intensity coming out. So this is a limitation, which is common to, of course, if the particle disappears, you cannot measure anymore. Okay, so the other thing that we discussed was that the piezo cannot be used, and we need to use the electrically tunable lens. By the way, the electrical tunable lens are very cheap, order $300-400, while the piezo is in the order of $5,000. So this is really very important also that we use this kind of thing. So the other thing, this is a single particle tracking. So if the particle is available, is visible in different, there are more than one particle, well, the system will not know which to track. And so that makes a problem. So still we have the approximation of following a single particle, but we can track in 3D while all the other techniques are not tracking in 3D. Okay, so let us look at one particular example. So in this example, this is the nuclear membrane of the cell. So this is the cytoplasm and this is the nucleus. And you can see there is some fluorescence coming out from this point. So we place an orbit here that is an orbit going in this direction. Okay, and going in this direction, if the particle is in that side, that is the center of the orbit, is in that side, will give more signal in one part. If it's the other side, it will be less signal on the other side. So why this is important? So first of all, we can track and not move. But if we do that, we will see that the X and Y in terms of seconds will move, because that structure will not be constant in position, but will move. So we cannot really track that particle and from that particle figuring out what happens. But we are interested in molecules that pass through the nuclear pore complex. And that is really the very important thing. And here, for example, you can see when you track the position, the average position, you see some molecules which stand in some particular part, 1, 2, 3, 4, 5, 6 position in this position. And every one of those positions give me an orbit such that says, at that angle, I see the particle. That another will be another particle, and the other will be another particle. So you can see that you get a correlation function. This is a correlation function that starts from 0, and then goes up, and then goes down again. And if you think about all the correlation function that you know from diffusion, never happens anything like that. It's always a function which is start high and goes low. Okay, so the other thing is that this is a work we did with Valeria Levi when she was a postdoc in my lab, that particles, which are here in red or in blue, are sometimes are connected with the nucleoli. If they are connected with the nucleoli, we don't measure really the motion of the particle, but we measure the motion of the nucleoli, which is where the particles are stored. So we did this figure. So we tracked this particle which is in blue here, and then we track a particle which is in red. And you can see immediately that the pattern that we get from the red, which could be associated with the nucleolus, but the pattern in the blue, but the pattern in the red is very different. So those two particles are following different dynamics. And that is how you can measure two particles at a time. Well, you do an orbit here, and then, for example, 10 orbits, and then you start to orbit the other one, and then you go back and orbit that one, and then you orbit the other one. So provided you know more or less what the particle is, and since the feedback is fast, you can always redetermine the position of the particle. Of course, you will lose the particle if the particle moves too fast, but this is not the case in this particular case. So with Valeria, we determine, for example, the motion of vesicle or organelles along the microtubules to measure the velocity and the number of motors they have and so on. And this is a work we did with Laura Estrada. And this is a very interesting work because this is what you see with your eyes. And then if you see, if you pay attention, you see that once in a while, a particle will stuck on the microtubule system and then it's moving. And how that can happen? So this is now one of those images. And then you can see that the particle can be found in many different positions and other particles are fixed. And now that, how can that happen? That can happen because the particle must be moved by light. There is no other source of energy here other than light. And this moved by light, and we figured that out, and we find a method that the particle can be moved directly by light, and so we can control the motion of the particle. Another thing that is very important, which was one of my comments to Fernando told today, is that we don't need to have an orbit which is exactly circular. We can oscillate the orbit. This is important because by oscillating the orbit, you will get closer or farther to the structure in which you want to figure out the shape. And by moving up and down the orbit or in farther, and you can get the shape of the object in 3 dimensions. And this is, for example, the shape of the object in 3 dimensions. And then you can see molecules moving along that part, which is shown here with this curve. So you can do many more things in this method, but you can also look at a 3-dimensional structure, which is very interesting, and you can measure at molecules moving in the 3-dimensional part. So this is also very interesting. So let us start to the final part of the talk, which is the tracking of a transcribing locus. So what we have here is a structure in which we have the pole tree, which is in blue, and then we have a promoter, and then we have when a certain part of the RNA is produced, we have a MS2 group EGFB, which is labeled with GFB, that will bind to that RNA. So we have this system, and then we can track a particle, and I will show you what is happening in this case. So first of all, if we increase the thing, we see that actually the loci is very complex and is not simple. And this very complex loci, which is in red here, you can see that the RNA, which is built, is available only in 1, 2, or 3, or 4 positions. And we call that the petals, and that's the center. So you have something which appears in that part of the orbit, the other things which appear in other parts of the orbit, and so on. So now we have a system in which we have simultaneous RNA expression in different sites, but the sites are very close one to the other. So the question is, how long will take one site to polymerize an RNA? How long will take the other site, and so on? And the rate of gene expression is very different, as you know. But here, what we're looking is a core, which is in red, and then the expression of the RNAs, which are in green in this part of the model. So this is now showing more or less what is happening. So we orbit in this region. So when we pass through to that, close to that petal, we will see an intensity, another intensity, another intensity, another. And you can see from this graph, which is time in second as a function of position along the orbit, that how much it will last. Each petal, then it will go to 0, and then will last, and so on. So we can measure the time it takes for a gene to be copied and to be expressed. And then we can make the statistic of that, and so on. And we can stop gene expression adding other things and so on. So this is what is happening. And then what is happening is that some genes, as you see, are active while the other are not. So which exclude that is the lack of energy in that position or in another position. And then we can calculate the autocorrelation function of the fluctuation that we see. And from the autocorrelation function, we can calculate the time it takes the gene to be expressed. So to our surprise, and the surprise to many people, we found that in this single loci, where several RNA are expressed, actually, we found that the elongation rate in base per second is about 20, on the average is 20, so that is the mean of all. But if you look, they are essentially identical, and doesn't matter what the cell is, and it doesn't matter what it is, the gene expression can start later on or not, but it's about the same. And if you look at the petals one by one, you see that also the gene expression, which is the addition of MS2 to that particular molecule, is about the same. So that points out that in petals which are very close to one to the other, and in sites which are all essentially the same position in the nucleus of the cell, the gene expression occurs at the same rate. And so we can then figure it out the motion of that part with respect to the nucleus, because now we have a different position and so on. I will not discuss that part, and I will say that simply that can be measured, and can be measured that initially there is a MSD, which is essentially the particle is moving, but as the time goes on, so the particle becomes larger and larger, it moves less and less because of the energy which is in So in this way now, we can finally have another information. When a gene is copied, so the RNA is copied, in which direction will go. And then that is in some way easy to do, because we can correlate that part to the orbit with another part of the orbit, with another part, with another part. So we can figure it out in which direction we'll go. And this is very interesting, because we see essentially the chromatin around, and the chromatin around is such that so when the particle is finished, it goes back, and then the next particle is produced. And then we can figure it out the direction in which the RNA will move. And for example, the RNA which is producing this part will always come away from that direction, but not from this direction. So evidently it cannot cross whatever is that chromatin space all around. So in conclusion, I say that the 3D particle tracking is based on fast and precise location of fluorescent particles within the orbit in 3D. This is allowed by the use of fast Fourier transform and circular scan orbit in feedback loop. Beside the on-the-fly information, the 3D particle trajectory, the following information are already available. So the shape of the particle, the lifetime of the particle, the spectrum, polarization, diffusion, binding, and kinetics. And this is the conclusion of my talk. And I hope I was able to carry on the information to you, to what 3D particle tracking can do. Thank you very much.